You're welcome to this series of tutorials at Medical Sciences Made Easy by Naphtali Muhumuza. And this session we want to talk about glycogenolysis. In the previous video we have looked at glycogenesis, how we convert glucose to glycogen to be stored in the liver and skeletal muscle. In this session we want to see how we can break this glycogen form to form free glucose which we can use to provide energy. So you want to see glycogenolysis, which is the breakdown of glycogen to form glucose so that we can utilize it to provide energy. And this process of glycogenolysis takes place in the liver, whereby the enzyme, the chief enzyme that catalyzes this reaction is known as glycogen synthase, or glycogen phosphorylase. So we see enzyme glycogen phosphorylase breaking down the glycogen to release glucose 1-phosphate. And how does it do it? We see this glycogen phosphorylase enzyme. For it breaks the alpha-1 4-glycosidic bond between glucose molecules by addition of the phosphate group. That's why we call it phosphorylase. Phosphorylase means it has phosphate groups. It keeps adding phosphate groups in the glycosidic bond, hence separating the glycogen. So we see this glycogen synthase coming and it starts adding phosphate group in alpha-1 4-glycosidic bond. And this synthase, if I can use this branch point, we see this glycogen synthase, it comes and adds a phosphate group and breaks it. It comes and adds another phosphate group. It adds another phosphate group, hence breaking the alpha-1 4-glycosidic bond. So this glycogen phosphorylase enzyme, it breaks down the alpha-1 4-glycosidic bonds by addition of phosphate groups. That whenever I add a phosphate group here, I cleave it off, I form glucose 1-phosphate. I come here, I cleave it off, I form another glucose 1-phosphate. I come and cleave this one off, I form another glucose 1-phosphate. That is the action of glycogen phosphorylase, whereby it breaks the alpha-1 4-glycosidic bond phospholytically or by addition of the phosphate group. But this enzyme, it functions until it reaches that carbon. If this is carbon number 1, carbon number 2, number 3, and number 4, it does not exceed at carbon number 4. So it can break these bonds up to this carbon, up to carbon number 4. Beyond carbon number 4, its action stops there. So it can add phosphate groups in the glycosidic bond up to carbon number 4. When it reaches at carbon number 4, it stops there, then we bring a deblanching enzyme. Brings a deblanching enzyme. That's right. It brings a deblanching enzyme, and this deblanching enzyme is the one that is going to come and cleave the 3. So we shall come and remove now these 3 remaining at the blanch point. And the enzyme that catalyzes this reaction is the deblanching enzyme. If we have a deblanching enzyme, and this deblanching enzyme, the first one is glucan transferase, whereby glucan transferase, for him, is a form of a deblanching enzyme which comes and picks the 3. Glucan means 3 molecules. It picks 3 molecules of glucose from carbon number 4 to carbon number 2, and he transfers it. That's why we call it transferase. And he transfers it to another branch point. So you see it, this glucan, or a 3 molecules being transferred to the adjacent. So we see it being added here by the glucan. This action is done by the glucan transferase. Glucan transferase enzyme, whereby it transfers when the phosphorylase stops at carbon number 4. It comes and picks the 3 glucose molecules and transfer them to the adjacent branch. That is the action of the glucan, which is a deblanching enzyme. Then after doing that one, we shall remain with only one more molecule of glucose at the branch point. We have only one molecule remaining after transferring the 3. And after transferring the 3, we know that this one molecule is joining at the branch point via alpha-1,6 glycosidic bond. So meaning we need an enzyme known as 1,6-glucosidase. We need a glucosidase enzyme. And this glucosidase enzyme is known as 1,6-glucosidase. It's the one that comes and cleaves this branch point at the branch point to release free glucose. So an enzyme, a deblanching enzyme, known as 1,6-glucosidase, comes and cleaves the alpha-1,6 glycosidic bond to release free glucose. And this free glucose is always ready for use. And what releases the free glucose at the branch point is 1,6-glucosidase enzyme. 1,6-glucosidase enzyme is the one that releases the free glucose at the branch point. And after removing this glucose molecule, the free glucose plus the glucose 1-phosphate formed, these guys which are formed here, they undergo it into this pathway whereby the glucose 1-phosphate is converted to glucose 6-phosphate by the same enzyme known as, it is called glucomutase, phosphoglucomutase. Phosphoglucomutase. This is being an isomerism reaction, we see it is reversible. So the same enzyme converts glucose 1-phosphate to glucose 6-phosphate by phosphoglucomutase enzyme to form glucose 6-phosphate. And this glucose 6-phosphate formed, it needs an enzyme. And this enzyme that catalyzes this reaction is known as glucose phosphatase. Glucose 6-phosphatase, found only in the liver. Meaning we cannot form glucose from glucose 1-phosphate in skeletal muscles because they lack glucose 6-phosphatase enzyme, found only in the liver. So this one is found only in the liver. So that's why we cannot utilize glucose 1-phosphate in skeletal muscles. In skeletal muscles, we can only utilize the free glucose that is released by a debranching enzyme known as 1,6-glucosidase. So that when in the liver we have glucose 6-phosphatase enzyme, it is the one that catalyzes the conversion of glucose 6-phosphate to form glucose. And this glucose which we have formed is the one that can enter into glycolysis and the Krebs cycle to provide energy. And that is how glycogenolysis occurs. Whereby in glycogenolysis, we need a glycogen phosphorylase. And this glycogen phosphorylase is the one that breaks the bonds by adding a phosphate group. And when it adds a phosphate group, we form glucose 1-phosphate. But it acts up to the 4 glucose unit, whereby beyond 4, we need a debranching enzyme known as glucan transferase, which transfers 3 molecules of glucose to an adjacent branch, then exposing 1 molecule of glucose, whereby this exposed 1 molecule of glucose at the branch point, we need a debranching enzyme known as 1,6-glucosidase, which cleaves the alpha-1,4 glycosidic bond to release free glucose. Then the glucose 1-phosphate formed, for it goes to the liver, whereby we convert it to glucose 6-phosphate. And this glucose 6-phosphate in the liver, we have an enzyme known as glucose 6-phosphatase, which converts it into glucose ready to be used to provide energy. This is glycogenolysis. Dear members, always remember to subscribe. And as we conclude this process, we need to know how it is regulated. This process is regulated. The regulation of this process, we need hormones, and these hormones, we need hormone like glucagon, hormone like epinephrine, and these two hormones, they stimulate. They stimulate, and how do they stimulate this pathway? Is that they stimulate this by adding phosphate or by phosphorylating glycogen phosphorylase. So when they add phosphate groups on glycogen phosphorylase, it becomes active. And how do we add phosphate groups? In endocrinology, we see that these guys, when this is the receptor, the G-coupled receptor, and a hormone binds, it comes and it activates glycoprotein or G-glycoprotein, which ends with adenyl cyclase. And this adenyl cyclase is the one, when activated, it is the one that hydrolyzes ATP to cyclic AMP, which is the second messenger. And this cyclic AMP is the one that activates protein kinase A in the cytoplasm of the cell. And this protein kinase A is the one that starts to add phosphate groups, is the one that phosphorylates this enzyme. So what glucagon and epinephrine do, when they bind on the G-coupled receptor, they activate adenyl cyclase enzyme, which hydrolyzes ATP to cyclic AMP, and this cyclic AMP increased levels of cyclic AMP activates protein kinase A in the cell, which has phosphate groups, and starts adding them to this enzyme known as glycogen phosphorylase, hence becoming active, stimulating the breakdown of glucose. And in case we don't want to break down glycogen, when in absence of these hormones, or in presence of insulin, for it inhibits the conversion. It inhibits, and then we cannot add phosphate groups. And then this one, when we don't add phosphate, it remains inactive. And then this is hormonal. Then allosterically, it is inhibited by increase in ATP. When there is increase in ATP, means we don't need energy, we need to store. So it inhibits this process. Increase in ATP inhibits this process. Thank you so much for listening. Always remember to subscribe and share your comments. Thank you so much. I don't know if they continued. I don't like, I'm going forward. I know it can show, but